Welcome to this lecture on principles of ecology. Today, we are going to explore the fascinating topic of community characteristics. Whenever we step into a forest, a grassland, or even a small garden, we are looking at a complex web of life. But how do we measure and understand this complexity? By the end of this video, you will understand the fundamental traits that define ecological communities and how we can mathematically calculate their health and diversity. Let us dive in. To begin our journey, let us understand what an ecological community actually is. Think of a bustling city neighborhood. It is not just made of one type of person. You have bakers, teachers, doctors, and mechanics all living together and interacting daily. If a neighborhood only had bakers, it would not function very well because essential services would be missing. An ecological community is exactly like this diverse neighborhood, but for nature. It is formally defined as an assemblage of interacting populations of two or more different species occupying the same geographical area at the exact same time. They share the same physical space and interact constantly. Now that we understand what a community is, let us look at its first major characteristic: species richness. This is perhaps the most straightforward metric ecologists use. Species richness is simply the raw count of different species present in a specific area. If you walk through a forest and count 10 different types of trees, your species richness for trees is 10. It doesn't matter if there are 1,000 oak trees and only one pine tree. As long as the species is present, it adds exactly 1 to the richness score. It gives us a baseline inventory of biodiversity. While counting species is helpful, it does not tell the whole story, which brings us to our second characteristic: species evenness. Evenness measures how equal the population sizes of these different species are relative to one another. Imagine two forests, both with 10 species of trees. In the first forest, every species has roughly the same number of individual trees. This is high evenness. In the second forest, one species makes up 99% of all the trees, while the other 9 species are incredibly rare. This is low evenness. A truly healthy ecosystem typically requires a balanced distribution. This idea of one species taking over leads us directly to our third characteristic: species dominance. Species dominance identifies the specific species that has the most biomass, or the greatest ecological impact on the environment. In our previous example, the tree species that made up 99% of the forest is the dominant species. Dominance is the exact opposite of evenness. When dominance is high, evenness is low. Identifying the dominant species is crucial because this single species often dictates the living conditions, available resources, and overall structure of the entire ecological community. So, why do we go through the trouble of measuring richness, evenness, and dominance? Understanding these community characteristics is absolutely vital for wildlife conservation and environmental management. You cannot just count the total number of trees and assume a forest is perfectly healthy. By measuring these specific traits, ecologists can detect early warning signs of environmental degradation, pollution, or climate change. For example, if a coral reef suddenly shows a massive drop in species richness, scientists know immediately that a stressor, like ocean acidification or warming, is damaging the ecosystem. These metrics make the invisible visible. To mathematically capture the essence of a community, ecologists frequently use the Shannon-Weiner Diversity Index. This formula is incredibly powerful because it combines both species richness and species evenness into a single numerical value. Let us break down the formula. H prime represents the final Shannon Diversity Index score. S stands for the total number of different species, which is our richness. The variable pi represents the proportion of individuals belonging to a specific species. We also use the natural logarithm function. The negative sign at the very front is crucial because the natural logarithm of a decimal fraction is always a negative number. Multiplying by a negative makes our final index a positive, readable value. When species increase or become more evenly distributed, this score goes up. Now that we have our formula, let us see it in action with a concrete example of a perfectly even community. Imagine a small garden with exactly 50 daisies and 50 roses. We want to calculate the Shannon Diversity Index. First, we find the total population, which is 100 flowers. Next, we calculate the proportion for each species. Since there are 50 of each out of 100, both proportions are exactly 0.5. We plug these into our formula, multiplying each proportion by its natural logarithm, which is roughly negative 0.693. After doing the math and applying the negative sign, we get a final index of 0.693. This represents the maximum possible diversity score for any two-species community. Let us contrast that perfectly balanced garden with another example to see the impact of dominance. Consider a different garden that also has 100 flowers and 2 species, but it has been taken over by weeds. There are 99 dandelions and only 1 tulip. Our total population is still 100. However, our proportions are now 0.99 for dandelions and 0.01 for the tulip. We plug these highly skewed proportions into the Shannon-Weiner formula. The natural logarithm of 0.99 is a very small negative number, while the logarithm of 0.01 is much larger. After multiplying and adding them together, our final index drops drastically to 0.056. Having calculated both scenarios, let us take a moment to compare these diversity scores. Our first garden gave us a score of 0.693, while our second garden gave us a score of 0.056. This is a massive and undeniable drop in overall diversity. Even though both gardens have exactly 2 species and 100 total individuals, they are fundamentally different ecosystems. The extreme dominance of the dandelions makes the second community mathematically and ecologically much less diverse. This perfectly illustrates why counting species alone is never enough to understand an ecosystem. To truly cement this concept, let us look at a visual representation of high versus low evenness. On the left side of this diagram, we see community A. It has high evenness and high diversity. Notice how the oak, maple, pine, and birch trees are equally distributed, with exactly 5 of each. The ecosystem looks balanced and resilient. Now look at community B on the right. It has the exact same 4 species, but it suffers from low evenness. The oak trees completely dominate the landscape with 17 individuals, leaving only 1 of each other type. Visually, it is obvious that community B is less diverse, and our mathematical index perfectly captures this reality. We have covered a lot of ground today, so let us summarize our key takeaways. Community characteristics are the essential metrics we use to understand the complex web of life in any given area. We learned that a healthy community requires not just species richness, but also species evenness, avoiding extreme dominance by any single group. Tools like the Shannon Diversity Index allow ecologists to quantify these traits objectively. Mastering these concepts is your first major step toward understanding how entire ecosystems are structured, maintained, and protected. Thank you for watching.